Thank you, Roger, for that most general introduction. But let me start out by saying, unlike science cafes, this is not meant to convey any new research results. This is meant to be entertaining. And so if you are here to learn something new, I notice there are people standing. This is a good time to leave. I will try to give you some hints about what I think are some of the highlights in the history of X-ray microscopy. Ben Feinberg points out that even I am not old enough to have been there at the beginning. But I will want to start with the year 1895, when Wilhelm Conrad Röntgen, who was at that time senior professor at the University of Würzburg, first observed X-rays. And it happened, in fact, on November 8th of 1895. And as it should be, he then spent the next 7 weeks working hard. Actually, he kept it all secret during that period of time. He did a number of experiments. He recognized very quickly that you can use this new kind of radiation in order to see bones in hands, can see metal objects in a wooden box, and he came to the conclusion that lenses and mirrors don't work. And so he then wrote up his paper. This is a paper that you will see has had quite a resonance. And on December 28th of 1895, he submitted his manuscript for publication to the Sitzungsberichte der Physikalischen Medizinischen Gesellschaft in Würzburg. I don't think that would count as a high-impact publication by today's standards, but it was the local scientific, the journal of the local scientific society. And, well, you know, in 1895, there were no phones, no fax, no email, no airplanes. You would imagine that it was a leisurely time when things didn't happen very fast, but 5 days or 4 days after he submitted his manuscript, he received the reprints of his article, which he sent out with some photographs to his friends. 4 days later, there was a front page story in the Viennese newspaper, Die Presse. 3 weeks, less than 3 weeks later, Röntgen gave the one and only public lecture and demonstration about his discovery. That same day, Nature, you know, Nature never accepts anything to be published that has appeared anywhere else. They published the English translation of the paper, and a week later, Röntgen was invited to Berlin to see the emperor and received a major decoration at that time. A couple of weeks later, Science reprinted the same paper along with 3 brand new original papers on the subject that were a result of the original discovery. One from Pupin in Columbia, one from Frost at Dartmouth, and one from Goodspeed at Penn. And a month later, there was a news brief in Science which said that the work of a large portion of the physicists of the world seemed to have contributed but little to the results published by Professor Röntgen. So in 1896, there were over 1,000 papers published on the new kind of radiation. Already all applications in medicine and industry have started. And furthermore, people started to recognize the dangers of dealing with X-rays. Röntgen had to wait another 5 years to get the Nobel Prize because Mr. Nobel was still alive in 1896. And the prizes had to be established, but he got the first one. So just to jump ahead 50 years, by that time in the 1940s, electron microscopy was being developed. And so the question was, is there a reason to think about X-ray microscopy, given the fact that there are all these other kinds of microscopies available? It was already recognized that transmission electron microscopy is limited in terms of the thickness of the sample that it can explore. And for unstained biological samples, the contrast was relatively poor, while in the case of X-rays, it was recognized that this was short wavelengths and penetrating radiation, so it didn't have some of the limitations of electron microscopes. And furthermore, absorption edges provide good contrast. It was recognized that between the carbon and oxygen absorption edges, water is relatively transparent compared to other kinds of material. The name water window was already coined before then. And furthermore, it was also recognized that X-ray fluorescence is a sensitive tool for trace element analysis. And to the extent that you could develop X-ray microscopes, you could map trace elements in samples with unprecedented resolution. However, there was no suitable X-ray optics. So let me go on to review a bit the development of X-ray optics. It goes back to 1918 and the work of Albert Einstein. This is the entire article of Albert Einstein from the Verhandlungen der Deutsche Physikalische Gesellschaft from 1918, where Einstein points out that the index of refraction in material for X-rays should be slightly less than 1, and therefore it should be possible to create total external reflection, and therefore there is the possibility for grazing incidence mirrors. In fact, grazing incidence was demonstrated by Compton some decades later, but it did not directly lead to anything like optics until a fellow by the name of Paul Kirkpatrick got into the act. Professor Kirkpatrick was a professor at Stanford, and he became interested in the possibility of building grazing incidence optics for imaging X-rays. And in fact, he published this article in the 1949 issue of the Scientific American entitled The X-ray Microscope. And the subtitle was, "It would be a big improvement on microscopes using light or electrons for X-rays combine short wavelengths giving fine resolution and penetration. The main problems standing in the way have now been solved." You should expect a microscope coming off the production line tomorrow. One of the figures in this article was one that you recognize today as the Kirkpatrick-Baez configuration of grazing incidence optics, where you focus in one dimension with one mirror and with the other dimension with the other mirror. And we know, love, and use this arrangement. However, Kirkpatrick did not recognize that this configuration is not an image-forming optic. It is perfectly good for creating microprobes, but he was hoping to create enlarged images, which led to some frustration. And so subsequent to Kirkpatrick's efforts, there was a period which I refer to as the Dark Ages. This is between 1950 and 1972. However, there were some workers working in the field, including a fellow by the name of Vernon Coslet, who, along with his collaborator, Mr. Nixon, published a book called X-ray Microscopy. This came out in 1960. And he described contact microscopy, projection microscopy. With projection microscopy depended on a microfocus X-ray tube creating divergent X-rays. And if you put an object in the divergent rays, you could record an enlarged image on a photographic plate, or describe absorption and emission microanalysis. But there was not a whole lot of following that, or follow-up that followed it. There was another guy who was involved at the time called Theodore Hall. He produced a book with his collaborators called X-ray Microscopy in Clinical and Experimental Medicine, and a review article on X-ray microanalysis in biology. But the reason why I mentioned to you is because some decades later, I learned from the front page article in the New York Times that Theodore Hall has been found out to be the second Soviet spy in the Manhattan Project in Los Alamos during the war, and he was a fugitive in Cambridge, England, working with Coslet, and it turns out that Hall and Coslet and Klaus Fuchs were all good friends. Coslet was a member of the Communist Party, and after he died, there were indications that he may also have been a Soviet spy. Now, at the same time, there were some developments, other developments in X-ray optics, in that Ora Myers and Albert Baez during this period have suggested that zone plates could be used to focus X-rays. And, you know, zone plates, we all know and love. These are diffractive optical elements, and you depend on the fact that those X-rays coming through alternate zones have a fast length to the focus, which is an integral number of wavelengths. And it's easy to show that the resolution of a zone plate, if coherently illuminated, is comparable to the finest zone that you can fabricate. Now, zone plates were not discovered in the second half of the 20th century. Zone plates were first described by Lord Rayleigh. Lord Rayleigh wrote about them in his notebooks, and his notebooks are all available, but didn't think it was interesting enough to publish. The first publication about zone plates came a few years later by the Swiss physician by the name of Sauvé. And in 1888, Lord Rayleigh published an extensive article on light in the Encyclopedia Britannica, and there he describes the fact that if you, instead of making alternate zones absorptive, you make them phase shifting, you can improve the efficiency with which light is collected to the first order focus. And in 1898, R.W. Wood published an article in the Philosophical magazine showing that he can take his camera, take off the lens, and put one of these phase zone plates instead and take nice landscape photographs and so on. And this rather poor-looking zone plate that you see on the right is a reproduction from that 1898 article in the Philosophical magazine. Now, coming back to the second half of the 20th century, after Ora Myers and Albert Baez suggested that zone plates would be useful for X-rays, the first person who tried to make zone plates for high-resolution imaging was a professor at Tübingen by the name of Millerstedt, and he was able to reduce a larger size zone plate so that the finest line width was a micron. But the real hero in this effort was Günther Schmal, who was at the time a professor at Göttingen, and he used ultraviolet holographic techniques to make zone plates with the finest line width of less than 1/4 of a micron. It was shortly after that that my friend and collaborator David Sayer wrote an article suggesting that he was working for IBM at the time, and he pointed out that the newly developed techniques of electron beam microfabrication would be a suitable technique to push this to even higher resolution. At the same time, in 1972, was the first time that the problem of X-ray sources has had a leap forward with the first application of synchrotron radiation to X-ray microscopy. This did not involve any major optics, although there was an elliptical mirror that was used to focus the zone plate onto a pinhole in a gold foil. This micron-sized pinhole was used to define a microfocus, which then determined the resolution of this instrument. They used Horowitz and Howell at the Cambridge Electron Accelerator, scanned their samples with this micron-sized probe, and detected either the transmitted radiation or fluorescence radiation, and demonstrated that in transmission, an electron microscope grid could be imaged with micron-sized resolution, or by using fluorescence, they were able to find little sulfur granules or a silicon whisker by using the characteristic X-rays. Notice the scale bar here is 50 microns and here it is 10 microns. But the first person to put both zone plates and synchrotron radiation together was Günther Schmal, who, along with his collaborators, built the first synchrotron-based X-ray microscope. This was operating at the Daisy synchrotron. Those were the days before storage rings. And this is from his paper published in 1976 in Applied Optics. So you see that it already involves a condenser zone plate, a microzone plate, and the microzone plate was used to form an enlarged image that they detected on a photographic plate, of course, at the time. And this is just an image from that paper showing a biological sample and demonstrating that the imaging works. Schmal then went on from the Daisy synchrotron to some early storage rings, first in Orsay in France, and then installed new generations of microscopes at Bessy. And of course, Bessy is still one of the major places where this kind of work is done. We got on to doing scanning microscopy as soon as the National Synchrotron Light Source was commissioned at Brookhaven in 1982-83. This is my former student, John Kenny, adjusting our very primitive piece of apparatus. And in fact, already in 1985, those of us active in the field at the time, Malcolm Howells, David Sayer, Günther Schmal, got together and wrote an article in Physics Today reminding people that, you know, X-ray microscopy is great and now all the problems are solved. It was a rather optimistic and enthusiastic article which showed some early pictures, but it suggested that one should be able to get down to about 10 nanometer resolution using either full field or scanning microscopy, and by doing diffraction imaging, maybe one should get to about 1.2 nanometer resolution. We are still on our way to get there, but we were interested in getting to three-dimensional imaging. We were talking about using undulators. So it was a lot of enthusiasm, and well, it took a little while to get there. But that sort of, in my mind, opened what I would describe as modern times, which was sort of the next decade, and that brought in lots more players, a lot of dedicated synchrotron storage ring light sources, improved zone plate optics, electronic detectors to replace photographic film, and people started to think about going away from just simple two-dimensional snapshots to either do tomography to get the third spatial dimension, or to do energy scans for spectral microscopy or X-ray fluorescence imaging, or to make movies where the third dimension would be the dimension of time. And along these lines, of course, we had to start really thinking carefully about radiation damage limitations. Now, right around that time in the mid-1980s, David Shirley was pushing hard to build the ALS. And he and Klaus Berkner came to the logical conclusion that in order to be able to justify and to have the infrastructure here, they have to build up X-ray optics in Berkeley. And so they established the Center for X-ray Optics, which has had a remarkable history. They recruited Dave Atwood to run the Center for X-ray Optics. Dave Atwood, in turn, recruited Eric Anderson, and the consequence of that was that well before the ALS was a working light source, Eric was able to make use of the nanowriter to build zone plates with 30 nanometer outer line widths, an order of magnitude better than what Günther Schmal had been able to make by holographic techniques. And this is still the way to do it, and the Center for X-ray Optics is still the place for making zone plates. There were also, to give you another anecdote, other efforts. NASA got involved in X-ray microscopy, and in 1992 they named Richard Hoover inventor of the year for the invention of the X-ray microscope, and they advertised this microscope as one that would have a resolution so high that it may produce detailed images of the building blocks of life, tiny DNA molecules. Now, this was not a flash microscope. This was going to be an imaging microscope, and so some of us wrote off to the director of NASA saying that this is, radiation damage is something that this doesn't seem to pay any attention to. We got 6 months later a response from the Inspector General of NASA that indicated that they concluded that no laws were violated. We just sent a short note back saying, except for the laws of nature. But no one has heard about this microscope since, so it hasn't bothered anyone very much. Now, here the person you see here is a fellow who many of you know. This is Harold Adem, and he was a graduate student and a postdoc with me at Brookhaven. And one of the things that he did was to get us involved in looking at the different components of polymers. And basically this was looking at a blend of polypropylene with a copolymer of polystyrene and acrylonitrile. And so when we looked at the model samples, it was very clear that the polystyrene acrylonitrile had a very bunch of very pronounced resonant peaks near the carbon absorption edge, while polystyrene, which is shown dashed, shows a very different spectrum. So by going to image at 285 eV, where the copolymer had a large peak, we got an image like this, while when we went to 288 eV, where polypropylene absorbs more, we got an image like this. So it was very clear that combining X-ray microscopy with X-ray spectroscopy is a very powerful tool to develop. And of course, that has been a major use of X-ray microscopy ever since. Now, during this following period that I would describe as the Industrial Revolution, what has happened was that things developed beyond where a professor and a bunch of graduate students got together and built a gadget, and engineers got involved to do things properly. And so better sources, better microscopes came online. There were a large number of third-generation light sources that were built. That's when hard X-ray microscopy got its start at the APS, at ESRF, at SSRL, and so on. Better optics became available, not just zone plates, but compound refractive lenses, corpettic bias, microprobes. One got away from very simple detectors to have CCDs and other area detectors, and even a company got founded that made it possible to have microscopes for every lab. You could buy your own. And so it was also the time when the ALS started operating, and I believe the first microscope to become operational was XM1, the Center for X-ray Optics microscope, starting up when the ALS started up, and it was operated for a number of years by Werner Meyer-Ilse before his untimely passing due to a major automobile accident. And one of the first publications coming out of that already in 1997 was this set of really remarkable images of malaria-infected red blood cells, which was published by the Proceedings of the National Academy. Not long after that came the first scanning transmission X-ray microscope at the ALS, and the first publication from that I found was by Tony Warwick, Harold Ade, Adam Hitchcock, Howard Padmore, and collaborators in the Journal of Electron Spectroscopy and Related Phenomena from 1997. They were looking at a slice of polyurethane foam and found that they could image it at high resolution and find the spectra from the different areas, so they could pinpoint what chemical components were present at the different places. But to make the scanning microscope enter the industrial area required a careful engineering approach, which was done by these characters. This is David Kilcoyne and Tilak Tilisak, and I believe that this is Peter Hitchcock, who built the first scanning microscope of the type that then became a prototype, which was then marketed by Axel and made available to the Canadian light source, this light source, and elsewhere. But the more recent X-ray microscope that came on board in Berkeley was the National Center for X-ray Tomography. This is one that Caroline Larabelle and Martin LeGros built, and that started operation in 2007, and it is now a very successful and productive microscope for biomedical application at the ALS, on the ALS floor. The more modern application of XM1 is now operated by Peter Fisher and his team, and the zone plates are built by Wei Chow and his collaborators, and it is primarily used in looking at magnetic vortex dynamics. So with this overall review of history, I want to give you a few examples of some of the things that go farther away from the Berkeley-centric approach and show you some of the examples from hard X-ray microscopy from other labs that I have found to be particularly compelling. This comes from SSRL, where Piero Pianetta and Joy Hoyster have built up a microscope which basically is was purchased from X-Radion and installed, and they, as well as many other people, have used the microscope to try to understand how to make lithium-based batteries better. And in their case, the cathode of the battery was made of nickel, which in the charging-discharging cycle cycles between metallic nickel and nickel oxide, where what you see in red is nickel oxide, what you see in green is metallic nickel, and blue is shown in mixed states. So as you go through the charge-discharge cycle, you can see that you go from the nickel oxide to the metallic nickel and back. Now, what you can do on a micrograph, at every point you can check at a point what the spectrum is, and at other point what the spectrum is, and you can compare that with the standards of the pure materials, where red is nickel oxide, green is nickel. So at this point you deal with 80% nickel oxide, here you deal with 81% nickel metal, and that's shown with these different colors. But what I want to point out, if in fact this is going to cooperate with me, is that you can take a tiny piece, this is probably about 5 microns across, you can take a tiny piece of this cathode material and do a tomographic reconstruction with chemical sensitivity, where again red is nickel oxide, green is metallic nickel, and blue is the mixed state. And so you can do this with significantly sub-micron resolution in 3 dimensions and with chemical sensitivity. In a completely different approach, people at the Advanced Photon Source built up the capability of using X-ray fluorescence analysis at high resolution and in 3 dimensions. And this is work by Martin de Jong and collaborators imaging a diatom-type marine organism, where you can see that the spatial scale here is 3 microns. And if I can get this one started, you are going to see the different components of this microorganism with all the different elements present with their concentration down to a milligram per cubic centimeter, and the whole architecture of the object is clearly displayed. And this I see as one of the real growth areas for X-ray microscopy as we go on in the next decade of development. So progress continues. There is further progress on sources. There are new storage rings like Petra 3 and NSLS2. You can buy yourself a very high brightness liquid metal jet source, or you can go to an X-ray free electron laser. And of course, there are plans and construction projects for multiband achromats, such as MAX4 in Sweden, Sirius in Brazil, and plans at the APS, at ESRF, and maybe I should mention that we have plans here at the ALS to convert the current light source into a very high brightness, much more coherent source, which will be terrific for microscopy. There is continued progress on optics. In order to make zone plates more efficient for hard X-rays, people have found ways of stacking zone plates to make them thicker. There are ideas of building multi-layer Laue lenses or compound refractive lenses in order to get high efficiency hard X-rays with very high resolution. There is progress on detectors, which is extremely important. You can now buy area detectors which are fast, efficient, and available. There is Peter Dennis's and his team's progress on fast readout CCDs, and I will show you examples of its use. For fluorescence detector, there are silicon drift detectors, and Peter Siddons and collaborators at Brookhaven have developed the Maya detector, which has hundreds of elements of energy-sensitive X-ray detectors, and pixel array detectors are also beginning to become available. And there is progress on instrumentation with all sorts of new types of microscopes, the availability of doing imaging at liquid nitrogen temperature to be able to acquire and analyze the images very fast. And also there is development in terms of the industrial base. X-Radio was purchased by Zeiss, and so you can buy all sorts of microscopes from them. Axel was acquired by Bruker, and you can buy all sorts of microscopes from them. And so just one example of the new type of microscopes that I wanted to mention is the one that is being built here as part of the Maestro project by Eli Rosenberg and his team, and that is to extend angle result photoemission spectroscopy to one where you can do it at high spatial resolution, maybe down to as small as 50 nanometers. But in terms of the worldwide availability and popularity of microscopes, I made a plot from the Web of Science. If I just type in X-ray microscopy, and I ask how many hits do I get per calendar year, it starts out with something on the order of a few dozen around 1960 when this book was published, to something on the order of 45,000 last year. Now, don't take this seriously. X-ray microscopy in the Web of Science means that the article has something about X-rays and something about microscopes, okay? But the fraction of these that involve X-ray microscopy is pretty universal over the years. So the numbers are highly exaggerated, but the rate of increase, I think, is realistic. And it is due to a variety of effects, but it is not mostly from cutting edge. It is due to the original motivation to look at opaque objects. And one of the really popular places that people have been looking at, especially not with necessarily with synchrotron light sources, but with laboratory instruments, fracking has become a bigger industry because people can use X-ray microscopes to understand how fluid permeates porous rocks. And what is fracking if not fluid permeation of porous rocks? So to find the right fluid, the right pressure, the right kind of rocks, this is one of the major uses of X-ray microscopy. Looking at failures of computer chips by electromigration and the like. Watching paint dry, concrete cure, charging and discharging batteries, fuel cells, and all these sorts of things has become a very popular undertaking. So Here is an example of a laboratory microscope, which is being used to understand what happens when you are using fuel cells. This is a small piece. The scale bar is here. It is 7 microns across, and you will see that we are going to enlarge it in a moment. The thing is, you want to understand how the gases go through the structure of the fuel cell, but the fuel cell operates at roughly 800 degrees. And so one of the questions is, how does the structure of this fuel cell change as you go from room temperature to the operating temperature? So X-ray microscopy is willing and capable of quantifying the porosity as a function of temperature, the connectiveness of the porosity as a function of temperature, the total pore surface area, and so on. And so this is where much of the growth has come from. Now I would like to change topic for the last 15 minutes or so, and tell you about developments which are not dependent on better X-ray optics, and talk about diffraction-based imaging. The basic idea is that normally we use optics in order to image an object, create an image, but lenses have limitations. And the question as to could we overcome these limitations has been the subject of this work. So you can replace the lens by some sort of a detector. And what the detector sees is the diffracted amplitude coming from the sample, which, if the object is coherently illuminated, involves a speckle pattern. The problem is that if you put a detector here, it cannot record the complex amplitude, it can only record the magnitude. And therefore you lose the phase, and it is not possible to create an image on the basis of that without some prior knowledge. If you do have some prior knowledge, then it may be possible to find an algorithm that will provide a reconstruction and spit out an image. Now, the history of this idea has a whole lot to do with David Sayer, who over more than half a century has been pursuing this idea, starting with a paper in 1952, where he showed that if all you can do is record Bragg peaks from a crystal, there is not enough information to make a reconstruction on the basis of that alone. And if you could just record information between the Bragg peaks, that would be very helpful. In the subsequent decades, there was a lot of work done in algorithm development, which culminated in the paper by James Phenop in 1982, where he showed that prior knowledge of knowing that your object that you are interested in is limited in size and there is nothing around it, which he called finite support, would make it possible to use an algorithm which he called the hybrid input-output algorithm to make a reconstruction. In the meantime, David Sayer insisted that we try to find whether a non-crystalline object is going to give a diffraction pattern, which we were successful in doing in the 1980s. And then Henry Chapman, along with David Sayer and John Yao, who at that time was a graduate student, showed that, in fact, if you could record this diffraction pattern, then at least in the case of a model calculation, one could use the Phenop algorithm to create a successful reconstruction. And next year, experimental demonstration became available when we took an artificial object, a bunch of gold dots that were deposited on a silicon nitride membrane. This is the scale bar of 1 micron. This is the speckle pattern recorded, and a successful demonstration of the reconstruction was performed. Now, shortly after that, Malcolm Howells built a beamline here called 901, and the team from Livermore, Henry Chapman and collaborators, and John Spence's team from Arizona State University joined to demonstrate that you can extend this coherent diffraction imaging to three-dimensional samples at reasonably high resolution, where this was a tip of a scanning probe microscope where some 50 nanometer gold particles were deposited on this object, and there was a set of diffraction patterns, a set of speckle patterns collected at the beamline 901, and the hybrid input-output algorithm was used to make a reconstruction of this set of gold dots to demonstrate that this technique works in 3 dimensions as well. Coherent diffraction imaging, however, has a lot of challenges when applied to doing work using a synchrotron light source. It requires coherent illumination. Even the ALS, which is as coherent as you get, that is a relatively difficult thing to arrange. You need to make sure that your object is isolated. There is nothing around it. Now you might say, how do you do that? You somehow have to mount the object on something in the beam. Then the other problem is that you somehow have to make sure that your detector doesn't get any signal other than the diffraction from your isolated object. Very hard to do. Furthermore, the diffraction pattern is extremely intense right in the direction of the beam and gets to be very weak when you go off at high angles. So this turned out to be a rather daunting set of criteria, and relatively few people today are pursuing this at synchrotron light sources. But right around that time, another development took place, largely the work of John Rosenberg of Sheffield University, who built on ideas that were originally put together by Hagel and Hockey for electron microscopy in the 1960s. And so Rosenberg got together with people at the Swiss Light Source, Pierre Thibault and his collaborators, to demonstrate another form of diffraction imaging called picography. Basically, once again, you detect diffraction patterns, but the prior knowledge, the constraint on the reconstruction that you need in addition to the diffraction pattern in order to do the reconstruction is very different. What you do is you take diffraction patterns from overlapping spots on the specimen. And it is this overlap that gives you the information that makes it possible to do the reconstruction. And they beautifully demonstrated this, and this is a much more rugged form of diffraction-based imaging, and it has taken off like wildfire and is being pursued in a variety of places, but nowhere better than it is done at the ALS. And so what I'm showing you is material that I got from David Shapiro a couple of days ago. This is work done by him with Young-Sun Liu, Oleg Tilisak, David Kilcoyne, Marian Farman, Rich Salestri, Stefano Marcassini, Tony Warwick, Li Yang, and Howard Padmore. And it is once again attacking the problem of lithium-based batteries, which is a really outstanding problem today. But now look at the length scale. Here we are talking about an aspect of the battery construction which involves lithiated iron phosphate versus delithiated iron phosphate. In these lithium batteries, the migration of the lithium ion is the critical component. And you can see if you look at these nanorods that are here, that if this is just an optical density image, but here you see it with the chemical information where red is lithiated iron phosphate and blue is unlithiated iron phosphate. And you tell them apart because near the iron L absorption edge, the pure iron phosphate, which is this lowest curve that you can't see, has a peak at above 710 eV, while the lithiated iron phosphate has a peak which is closer to 708 eV. And so you can tell apart what is lithiated and what is not. And so this is a map of the lithiated iron phosphate, and this is a map of the iron phosphate. And you can do that even in 3 dimensions. This is a little granule of this battery material. The entire size is 80 by 80 by 20 nanometers. So the resolution is just a few nanometers across, and you have it with chemical speciation. You can use this technique to find at very high resolution in 3 dimensions what chemical component is where. And so this is done on the basis of the nano server, which is running at beamline 5321, and it depends both on the high-speed CCD that was developed by by Peter Dennis's group, and on very high stability scanning stages that were built in preparation of this becoming the major occupant of the new beamline, one branch of the new beamline cosmic at beamline 7. And for the last couple of minutes, I want to spend time telling you about what free electron lasers can do for X-ray microscopy. Free electron lasers, of course, give you coherence without your doing a lot of work, and so that removes one aspect of the difficulty in doing coherent diffraction imaging. The pulses come in a few femtosecond time, and therefore you don't have to worry about radiation damage. On the other hand, you are going to destroy your object. You have to catch the diffraction pattern fast, and because you destroy it, you cannot do tachography because you can only get one shot from the sample. This was first demonstrated by Henry Chapman's group a decade ago at the Flash 3-electron laser in Hamburg, where they took a little microfabricated object, they hit it with the 3-electron laser, a single pulse of the 3-electron laser to collect the diffraction pattern. They were able to reconstruct it in a way that mimics the scanning electron microscope or the transmission electron microscope image. And if you record the next shot, you see that the thing is gone. Basically, what you are looking at is an empty frame. Now, the reason why people have been so excited about that was the suggestion that if you could do that on individual protein molecules, which are such that they could not be crystallized, maybe you could collect the diffraction pattern from these protein molecules and do a reconstruction without having to crystallize. But it was demonstrated by Neitze et al. In 2000 that you would have to go faster than 50 femtoseconds in order to outrun the inevitable blowing up of the molecule. And so the idea has been that you could inject these individual molecules into the free electron laser beam, and collect the speckle pattern each time round. And so what you would do is to take maybe a few million of these, and then because the molecules are in arbitrary orientations, you would have to find the ones of the same orientation and then average these, and then take all these averages of different orientation to fill the three-dimensional diffraction space, and then you could use the hybrid input-output algorithm to do a reconstruction. Now, as it turns out, this dream, as of today, is still a dream. People have not been able to collect diffraction patterns at 3-electron lasers intense enough to be able to classify them and find out what the orientation is. However, what people found out is that in many cases, if you cannot get one molecule, you can get nanocrystals of some materials. And to me, the most compelling example is the nanocrystals of catepsin B, which are involved in the sleeping sickness disease, which is spread by a protozoa, a parasite, and which affects millions of people. And people have been trying to find out whether this protein is suitable for developing drugs to treat the disease. And based on some millions of these shots of 3-electron lasers on the injected nanocrystals of this protein, they were able to collect some 300,000 hits in an 8-hour time at the LCLS to generate a diffraction pattern that you can see here. And on the base of the diffraction pattern, they were able to reconstruct the molecular structure, which to me is an amazing achievement. So my time is pretty much up. It's about time that I come to some conclusions. I hope I convinced you that nowadays there are lots of microscopes with lots of applications and lots of exciting developments in picography with three-dimensional imaging and chemical sensitivity, X-ray fluorescence mapping of trace elements, nanocrystallography of 3-electron lasers, nano-angle result photoemission. I did not have a chance to talk about nanodiffraction to study stress and strain at the sub-micron level, nanomagnetism, and so on. And of course, all of these techniques will be helped with more brightness and more coherence at future light sources. But you have to keep in mind the limitations. There is radiation damage, and I urge you to, if you're interested, take a look at the paper by Malcolm Howes and collaborators on how that limits things. And keep in mind that X-ray microscopes are not the only game in town. There have been major developments in light microscopy, electron microscopy, and scanning probe microscopes. And in fact, there have been a number of Nobel Prizes in the last decade having to do with those other alternatives. So we have to concentrate on those applications for which X-rays are unique. And I want to apologize for having left out many important milestones, many important contributions, many humorous anecdotes, and I want to comment that, you know, I think if Mr. Röntgen could see what has happened, he would be rather pleased. I would like to thank my colleagues here at the ALS and others whose results I have shown, and of course, we are all helped by the Department of Energy, and I thank you for your attention.
Thank you. Maybe, I know some people have to leave, but if there are burning questions, prevail on you to answer. I have one about holography. Why is it, will we in the future be using sort of conventional holography
Yes. Holography, of course, also benefits greatly from coherence and brightness. And if I would have had an hour and a half instead of an hour, I would have mentioned that as the kind of holography which has been shown to be particularly productive in terms of X-ray imaging is Fourier transform holography. This has been shown both at synchrotron light sources and at free electron lasers to make it possible to create very simply reconstructible structures which don't require iterative algorithms and other constraints.
We know it all. Okay, Janos, thank you again.